morning. Today we are going to talk about sensory neuroscience. We have been seeing the seminars on pain pathways and sensory neuroscience. What sensory neuroscience means? Sensory neuroscience is nothing but a biological mechanism by which sensory receptors convert the physical or chemical stimuli into electrical signals, and how these signals are transmitted along the neural pathways and how they are integrated and transformed into perception, behavior and decision making. So this is what sensory neuroscience means. So in pain sensory neuroscience, it's a subfield of sensory neuroscience that specifically examines how the nervous system detects, transmits and interprets the noxious stimuli that give rise to the experience of pain. So what are the basic mechanisms of pain? There are three mechanisms, transmission, transduction and modulation. There is a presence of noxious stimuli. So the stimulus events are converted to chemical tissue events. The chemical tissue and synaptic cleft events are then changed into electrical events in the neurons, and the electrical events in the neurons are transduced as chemical events at the synapses. This is the mechanism of pain. So what are neurons? So neurons are the basic structure of the nervous system, and there are three types of neurons that exist in our body. There are the sensory neurons, interneurons and the motor neurons. The sensory neurons will be like this. The interneurons is a neuron which connects between the neurons, and the motor neuron connects to the muscles. It supplies the information from the brain. Sensory neurons takes information to the brain. And neurons are electrically excitable, and it consists of soma, axon and dendrites. Soma is where the nucleus is present. It supplies a nutrient to the nerve cell, and the axon is, it can either be myelinated or unmyelinated, and the dendrites are the branching which connects with the next neuron. The neurons are connected with each other to form complex neural networks in our body, where the chemical and electrical signals are transmitted via specialized connections which are called synapses. So we can see how the nerves interconnect with each other, and this is called the synapse, where the axon terminal of one interacts with the dendrite of the other. Talking about axons, axons are all known as nerve fibers, which are main component of the neuron and functions to conduct the action potentials in a unilateral direction. So it conducts always in a unilateral direction from the dendrites to the axonal terminals, as well as from one neuron to other. So we have the myelin sheath here in this axon, and in between the myelin sheath we have the nodes of Ranvier, where the signals are transmitted. The depolarized and repolarized membranes we can see, and this myelin sheath forms the insulation so that the polarization or the electrical signals are not dissipated. So this forms the insulation so that the action potential travels in a unidirectional manner. And the presence of myelin sheath of an axon known as the nodes of Ranvier increases the propagation speed of impulses as they travel along the myelinated fiber via saltatory conductions. That is, generation of action potential at each nodes of Ranvier is saltatory conduction, and acts as an insulator to prevent the electrical impulses. As I said, it protects the nerve signal. So we can see how the nerve signal is transmitted. This is the saltatory conduction from one node to the other node. It transmits. And we have three types of nerve fibers: group A, B and C. When we talk about group A, these fibers are myelinated. It can be further divided into alpha, beta, gamma and delta. So A alpha fibers are proprioceptive in function, beta fibers are mechanoceptors. Proprioception is the position or something like that. Mechanoceptors is the pressure kind thing. A gamma is stretch receptors, and A delta are thermal and mechanical nociceptors. They are the smallest myelinated nerves and have a relatively fast conduction velocity of 30 meters per second. That is why the A delta fibers are the fastest conduction, and the nociception is so fastly conducted because it travels at the speed of 30 meters per second. And the diameter of the A delta fibers is about 2 to 5 micrometers and is responsive towards short lasting and pricking pain. So A delta fibers are short lasting and pricking pain conduction. We have the group B fibers, which are the pre-ganglionic nerve fibers of the autonomous nervous system. Autonomous side, we have sympathetic and parasympathetic nervous system. In that, group B fibers are present, and they are general visceral afferent fibers belongs to this group. Group C fibers, the nerve fibers at the dorsal roots, and postganglionic fibers in the autonomic nervous system. We have the preganglionic and postganglionic in the sympathetic and parasympathetic. You know that the preganglionic fibers are long in the parasympathetic, whereas the postganglionic is short. In sympathetic, we have the short preganglionic and the long postganglionic fibers. So in these fibers, we have the C fibers. And they are the main nociceptive fibers which carries the sensory information, assembling around 70% of the afferent nociceptive information, and which enters the spinal cord. The nociception of the C fibers are polymodal, which means they can detect thermal, mechanical and chemical stimuli. And 50% of these fibers express neuropeptides such as CGRP, which we have already seen, calcitonin gene related peptide, neurokinins and substance P. So these are the three types of nerve fibers, A, B and C. Talking about action potential. So the release of neurotransmitters at the axon terminal is triggered upon the entrance of calcium. So what happens when at the end of the nerve fiber, which is the axon terminal, there are calcium gated ion channels, and at that, when there is action potential which reaches the axon terminal, the calcium ion enters the axon terminal and it triggers the release of the neurotransmitters which are stored in vesicles, which is called in the axon terminal. These neurotransmitters come out and reach the synaptic vesicle cleft, which is the junction between the presynaptic and postsynaptic membrane. And there are two major potentials, the resting potential and the threshold potential, which are approximately -70 and -55 millivolts respectively. So this is important in the action potential. We need the resting potential and the threshold potential. There are more sodium ions accumulated outside the cell than the potassium ions inside the cell, and thus the resting potential of the cells is always negatively charged. The movement of these ions across the lipid bilayer of the neurons is strictly dependent on the ion channels. So when we see the action potential, we see one curve which goes above and then it comes below. So the shape of the action potential is always stereotypical, which means it is always the same. Which the amplitude and the time course of all the action potential occurring in the cells are the same. So this is the action potential which happens inside the nerve fibers. So the ion channels, when we talk about ion channels, we have voltage activated sodium ion channel, NAVs, voltage activated potassium channels, and we have this calcium channels. When the action potential travels to the axon terminal, the calcium enters into the presynaptic terminal through the voltage operated calcium channels, hence causing the synaptic transmission. So these are the important points we should note in talking about action potential. And how is the synaptic transmission happening? So when action potential reaches here, the calcium ion channels, calcium enters. These are the synaptic vesicles carrying the neurotransmitters, and they come near the presynaptic membrane, fuse with the membrane and release the neurotransmitters in the synaptic cleft here, where we find the postsynaptic membrane having all the ion channels. These neurotransmitters enter inside the postsynaptic membrane and then conduct the action potential. And this is the mechanism by which the synaptic transmission is happening. And the route of pain transmission, we have the ascending route, ascending system. And first what happens, the spinal cord, brainstem, medulla oblongata, midbrain and the cortical regions are the areas involved in the processing of pain. The dorsal horn of the spinal cord plays a crucial role in integrating multiple, we can see multiple inputs enter, and then including the primary afferent neurons and local interneuron networks, and is also responsible for the descending signals from the supraspinal center. So we have both the ascending as well as the descending. Ascending takes the sensation, descending modulates the sensation of pain. So the primary afferent nociceptors projects the neurons into the dorsal horn of the spinal cord, then to the thalamus, then to the somatosensory cortex through the spinothalamic tract, thus providing information of the intensity and location of the noxious stimuli. The spinothalamic tract is located in the white matter of the spinal cord and consists of two parts, the lateral and the anterior spinothalamic tracts. The lateral spinothalamic tract focuses on transmission of pain and temperature sensation, whereas the anterior spinothalamic tract carries information related to the crude touch firm pressure sensation towards the thalamus and the brain. And this is how we see the thalamic tract. It crosses contralateral side and then it ascends as the medial and lateral and the anterior spinothalamic tracts to reach the brain. The ascending information accesses the neurons of the periaqueductal gray matter. These two are the areas in the brain which try to control the pain mechanism, either to perceive the pain as higher value or lower one. So the neurons of the periaqueductal gray matter and rostroventral medulla that is found in the midbrain to integrate the information received from the higher centers of the brain, including the hypothalamus, amygdala and frontal lobe, as well as receiving the ascending nociceptive input from the dorsal horn. The endogenous opioid and cannabinoid systems and other neurotransmitters such as 5-HT and norepinephrine are heavily expressed through the PAG or RBM pathways. So these are the routes of transmission. And what are the pain-mediated intracellular effectors? We have the G protein coupled receptors, which is a large family of cell surface receptors that play a pivotal role in cellular communication and signal transduction. So this is the G protein coupled receptor. We have the extracellular and the intracellular. So the GPCRs are characterized by their 7 transmembrane helices which span the cell membrane, and the extracellular portion of the receptor binds to specific ligands, while the intracellular portion interacts with the G proteins to transmit the signal to the cell. So this is one of the intracellular transmission effectors. And the next is ligand-gated ion channels, which are ionotropic. So these are transmembrane proteins that facilitate rapid synaptic transmission by allowing ions to pass through the cell membrane in response to binding of specific chemical messengers such as neurotransmitters. So we can see the ion channels here. So this is the receptor and this is the messenger. When the messenger reaches there, the ion channel opens and there is exchange of ions either outside or inside. So this is the structure of the ion channel. So upon ligand binding, these channels undergo a conformational changes, which means there is a structural change, opening their pore to permit the flow of ions. What are all the ions? We have sodium, potassium, calcium and chloride ions, thereby altering the cell membrane potential and initiating a physiological response. So we know pain is a huge chunk of finances within the healthcare system, which is the main reason why all reach the healthcare system, and it needs continuous rehabilitation of patients with adverse pain sensations, which might reduce not only their quality of life but also their productivity at work, setting back the pace of our economy. So pain is a major area of healthcare system which needs to be addressed. Thank you.